Hello everyone. Today we are proceeding into microbiology part 4. In microbiology part 4, we will be dealing with viruses. First, we will take a look at the classification of viruses, their characteristics, and then we will discuss about the status of viral multiplication. And in the upcoming videos, we will discuss about bacteriophages, how they reproduce, the lytic and the lysogenic cycle. We will take a discussion on tobacco mosaic virus, retroviruses, and other infectious agents like viroids and prions. So let's proceed to the relationship of microbes, or just a recap. So as we have already learned, microbial world is broadly divided into organisms which are considered living, which includes bacteria, archaea, and the eukarya. And then the second one is the non-living infectious agents, under which comes the viruses, viroids, and the prions. We have already finished with the bacteria and the archaea, and now we are passing on to viruses. Virus, the word means toxin or poison. They are supposed to be the smallest and simplest forms of life on earth. They are submicroscopic, even smaller than the bacterium, and they are highly infectious agents which cannot grow outside a living host. So that is why these viruses are considered to be non-living. They become living only when they infect a living host. That is why viruses are known as obligatory intracellular parasites. Viruses are called obligatory intracellular parasites because they can live and reproduce only when they are inside a living host. So this you can get as a one-word question. Obligatory intracellular parasites, they are organisms or parasites which can grow and reproduce only within a living organism. So a little story about the discovery of viruses. Researchers were studying a particular plant disease known as the tobacco mosaic disease. The disease is characterized by stunted growth and mottles in the plant leaves. So the research team led by Professor Mayer was studying about what is causing tobacco mosaic disease. So he found that there were certain infectious agents which were transmitting the disease, and he found that the disease could be transmitted by spraying the sap from the diseased plant to a healthy plant. But he also knew that they were produced by some infectious agents which are very, very, very small, maybe smaller than the bacterium. And later on, Dmitry Ivanovsky, another scientist and his group, demonstrated that the sap was still infectious even after passing through a filter which was supposed to remove bacteria. So Mayer was supposing that the tobacco mosaic disease is caused by a very extremely small bacterium. But Ivanovsky proved that these infectious agents could not be bacteria because even after passing through the filter, the sap was still infectious. So later on, Martinus Beijering and his team ruled out the fact that the filterable toxin from the infected sap is a bacterium. How did they do it? They demonstrated that the infectious agents could reproduce inside a living host, and the sap from one generation of infected plants could infect the second generation, the third generation, and so on. So later, what he found was these infectious agents could not be cultivated in a nutrient medium like a bacteria, nor could they be inactivated by alcohol, which was supposed to be lethal to bacteria. Later, Martinus Beijering named this infectious agent as a virus. So who named the infectious agent as virus the first time? It was Martinus Beijering. And in 1935, Wendell Stanley crystallized the pathogen which is causing the tobacco mosaic disease to be the tobacco mosaic virus. and it is known as TMV, all in capital letters, tobacco mosaic virus. So this is a story about how viruses were discovered. Now let us get to know some of the characteristics related to viruses. Virus particles are very small. They are even smaller than a bacteria, and they multiply inside the living host only, and they cause diseases. The infectious particle of a virus is known as a virion. So don't get confused with this word. Virion is an infectious virus particle. Moving on to the classification of viruses. Viruses can be classified based on many different criterion. The first criterion is that they can be classified based on the type of the host they infect. They can be animal viruses, they can be plant viruses, or they can even be bacterial viruses. Let us get to know some of the virus groups. Viruses which infect plants are called the phytoviruses. Zooviruses are those which infect animals. Mycoviruses or the mycophages are viruses which infect fungus. Phycoviruses or the phycophages are otherwise called the algal viruses, and they infect algae. Bacterial viruses, commonly known as the bacteriophages, are viruses which infect bacteria. And there are also viruses which can infect mycoplasmas. So, based on the host, type of the host which the virus infect, they are named as phytoviruses, zooviruses, mycoviruses, phycoviruses, bacteriophages, or mycoplasma viruses. The most important and well acclaimed classification of viruses is by the International Committee on Taxonomy of Viruses, in short known as the ICTV, all in capital letters, International Committee on Taxonomy of Viruses. They classify viruses under 8 clusters based on 3 major aspects. So based on 3 major criterions, ICTV clusters the viruses into 8 clusters. So what are the 3 major criterions that they take into account for classifying the viruses? The first thing is the kind of nucleic acid or the genetic material that is present within the viruses. So the nucleic acids are genetic materials like DNA or RNA which is present in the virus. The second one is the strandedness of the nucleic acid. That means whether the DNA or the RNA present in the virus is a single-stranded molecule or a double-stranded molecule. And the third criterion they use to classify viruses is presence or absence of an additional envelope. So normally virus has got a protein cover and inside which you can see the genetic material. So in some viruses, apart from the protein covering, they also have an additional envelope. So ICTV or the International Committee on Taxonomy of Viruses uses these 3 criterions to classify the viruses into 8 clusters. Let's check them out. So viruses are broadly classified into DNA viruses or RNA viruses based on the type of nucleic acid present within, and the second criterion that is being used is the strandedness of the nucleic acid. So it can either be a single-stranded DNA virus, a double-stranded DNA virus, single-stranded RNA virus, or a double-stranded RNA virus. The third criterion that ICTV uses is whether there is an additional envelope present or not. That means viruses can be single-stranded DNA virus with an envelope, single-stranded DNA virus without an envelope, double-stranded DNA virus with an envelope, double-stranded DNA virus without an envelope. Or they can be single-stranded RNA viruses with an envelope, single-stranded RNA viruses without an envelope, double-stranded RNA virus with envelope, or double-stranded RNA without envelope. That means these are being clustered into 8 different clusters based on the three characteristics. What are the three characteristics that ICTV uses to classify viruses? They include the type of nucleic acids, the strandedness of the nucleic acids, and presence or absence of an additional envelope. You need to remember the classification of viruses based on ICTV, and ICTV is International Committee on Taxonomy of Viruses. Then there is another type of classification based on LHT system. LHT standing for Loff, Horn, and Tonier. So these are the three scientists working behind this system of classification, and it was put forth in 1962. So LHT system uses two additional characteristics for classifying the viruses. So the three characteristics that was used in ICTV is still being used in LHT system. They include the kind of nucleic acid, strandedness of the nucleic acid, presence or absence of the envelope. The two additional characters that LHT system considers include the capsid morphology. As I said earlier, viruses have a protein coat, and the protein coat is known as the capsid. So here, LHT system uses the morphology of the capsid, including size, symmetry, number of the building blocks which make up the protein coat, into consideration to classify the viruses. And the last one, the last category that it uses is the range of host, which all systems or hosts these viruses are capable of infecting. So LHT system uses additional two characteristics than the ICTV to characterize viruses. So altogether there are 5 categories that is being taken into consideration to classify the viruses into different groups. They include kind of nucleic acids, whether it is DNA or RNA, strandedness, single-stranded or double-stranded, capsid morphology, presence or absence of an envelope, and the range of host. So these are the characteristics that Loff, Horn, and Tonier uses in LHT system of classification of viruses. The infectious particle, as I said earlier, virus particle is known as a virion, so don't forget that word. Then I said virus particle or the virion consists of genetic material and a protein coat. Genetic material can either be DNA or RNA, and the protein coat is called a capsid. So that means virus is a capsid plus a nucleic acid, or you can say it as a nucleocapsid. So the nucleic acid within a protective protein coat, that is what the viral particle is all about. Then I spoke about capsid, that is the protein covering. The protein covering can vary from a very simple helical structure to very complex icosahedral types. Icosahedral meaning polyhedral or near spherical forms, and even very, very complex structures with tails or an envelope. So tails you can find in bacteriophages, envelope you can see in very notorious, highly infectious viruses. So here you can see a glimpse of the diversity of the viral structures. We will see it a little bit later. So let us get to know the words once again. The infectious virus particle is known as a virion. A virion or a viral particle comprises of two major elements. One is the genetic material of the nucleic acid. The second one is a protective protein coat called the capsid. So it is together known as a nucleocapsid, and the capsid varies in size, shape, and the structure. The capsid or the protective protein covering is made up of building blocks known as capsomeres. Protein subunits of a capsid in virus is known as capsomeres. And the major function of the capsids include it protects the nucleic acids from inactivation by nucleases of the host. Nucleases are nothing but enzymes which can degrade the nucleic acids. So nucleases are nothing but enzymes which can degrade the nucleic acid. So normally there is inactivating nucleases are produced by a host as a measure of immune system. So another function that the capsid does is It helps in introducing the viral genome into the host cell. How do they do it? By first adsorbing onto the surface of the host cell. So first they will come, land on the surface of the host, then they adsorb so that the cell does not move, and it injects the genetic material into the host cell. We will see about it a little later. Then also, these capsids are protective in function. That means it determines the antigenic specificity of the viruses, how far these viruses are infectious, how they can resist the activity of cells that are used to kill them. So all these is being determined by the type of the capsid. So capsids are nothing but the protein covering seen in the viruses. They are made up of protein subunits known as capsomeres. Major function is to protect the nucleic acids from inactivation by nucleases of the host. They also help in adsorbing to the cell surface and determine the antigenic specificity of the viruses. You can see here different types of capsid morphology. So this is how they look under the microscope. And this is just diagrammatic representation. What you are seeing here is tobacco mosaic virus, and this is an adenovirus. So this is a helical structure, and this is a polyhedral structure. And apart from the capsid proteins, some viruses have viral envelopes. We saw that earlier. And these membrane blocking the capsids give the viruses special properties. For example, they help the viruses to resist chemicals, as well as they give biological and antigenic properties to the viruses. And most of the time, these envelopes are seen in very notorious and highly infectious viruses. And the funny part is, these envelopes, which help them to resist the chemicals they use to kill them, are being derived from the host cell membrane. And these envelopes are also made up of viral proteins and glycoproteins. They also have viral proteins and glycoproteins. You can see here the example of an enveloped virus. The pink thing that you are seeing here, that is a nucleic acid. Then you can see a lilac purple colored thing, that is a protein coat. And this one, the gray thing with the purple pins, are the membranous envelope, and the pins are nothing but the glycoproteins, the projecting spikes. Okay, so this is an influenza virus. The most complex structures found in viruses belong to that of bacteriophages, or the ones which we simply call as phages. These are nothing but viruses which infect bacteria. So the structure of bacteriophages is such that the protein capsid is very complex, and mostly it has got a head and a tail part with all the tail fibers and the tail sheath and so on. And the nucleic acid is highly protected within this head. And the head capsid is icosahedral with 20 sites. So the T-even phages that infect E. coli or the Escherichia coli is a bacterium. And this is the structure of the bacteriophage which infects E. coli. So we will learn in detail the structure of bacteriophages in another class. Just to show the diversity of all the different types of viruses possible, the enveloped one, the influenza we saw a little earlier. This is the tobacco mosaic virus, the first virus to be identified. This has got a helical structure. And this is another virus, adenovirus, which is polyhedral in shape. Again, all these little beauties that you're seeing over here, these are all viruses. Don't go by beauty. They are all very highly infectious ones, and you must be very familiar with this one, the coronavirus, which is now giving us a lot of headache and causing the world pandemic. And herpes virus, influenza virus, smallpox virus, every one of them is highly infectious. Then moving on to the genomic structure. Genome is nothing but sum total of all the genes present within an organism. So where do we find genes? In as a genetic material. So in this case, in viruses, you can see a vast diversity in the genome structure. It can be difference in nucleic acid, the shape of the nucleic acid, or the strandedness of the nucleic acid. So nucleic acids can either be DNA or RNA, and it can either be single-stranded or double-stranded. So here you can see that based on the nucleic acid, the viruses can be divided into two, DNA viruses and RNA viruses. These DNA viruses can replicate using a DNA-dependent DNA polymerase, that is an enzyme, and RNA viruses replicate using an enzyme, RNA-dependent RNA polymerase. They can either be single-stranded or double-stranded. Then there are differences in the shape of the nucleic acid present. It can be linear in shape, it can be circular in shape, or in some eukaryotic viruses, you see more than one nucleic acid. That means it is segmented. So based on the shape of the nucleic acids, they can either be linear, circular, or in eukaryotic viruses, they are segmented. We will now pass on to how the virus multiplies, the steps in viral multiplication. As I said earlier, viruses are supposed to be non-living until they enter into a living host. So for a virus to grow and multiply, they first have to infect a living host. So let's see how the virus multiplies. The viral multiplication involves a series of sequential steps. So replication can take place only once it has entered into a living host. Why is it so? Because the virus as such do not have any biosynthetic machinery. The enzymes which are needed for multiplication or reduction of proteins, we do not have any such biosynthetic machinery. So how do they replicate? Once they enter into a living host, it uses the genetic material of the virus to take over the biosynthetic system of the host, and with the help of the biosynthetic system of the host, it replicates itself. So let's check out how this happens. So this pictorial representation gives you the sequential steps in viral multiplication. At first, here what you are seeing here is a bacteriophage infection. Bacteriophage infects bacteria. So in step 1, what the virus does is it comes and lands on the host cell. In this case, it is a bacterium. So the first step is attachment. The second step is known as penetration. What happens during penetration? The viral DNA or the genetic material from the virus is injected into the host cell. In third step, biosynthesis takes place. That means the viral particles are being synthesized. It includes replication of the genetic material of the virus as well as the production of the protein coat or the capsid. And in the fourth step, assembly takes place. That means the genetic material is packed inside the capsids and the virions are getting ready. And the final step is it kills the bacterial cell, host cell, and releases itself out into the world. So let's discuss it a little further. So the sequential steps in viral multiplication, it starts with the virus absorbing or attaching itself onto a particular host. How do they do it? With the help of specific cell surface receptors, which include glycoproteins or lipoproteins. Then in the second stage, it penetrates the membrane of the host cell and injects its genetic material into the host cell. In some cases, the virus as a whole, including the capsid, can enter into the host cell, and in some cases, only the genetic material is injected into the host cell. And then in the biosynthetic stage, what happens is it takes place in two different ways. So uncoating takes place, and the process by which the viral capsid is stripped off and it releases the nucleic acid into the host cell. So that is when the whole virus is entering into the host cell. But most of the time, the viral capsid is kept outside, left outside, and only the genetic material is penetrating inside the host cell. So whatever it be, once the genetic material of the virus is inside the host cell, it takes over the machinery of the host cell and starts biosynthesis. So what happens during this biosynthesis? Mainly genome replication takes place, as well as whose genome? The virus genome. And then it also starts producing hot proteins, enzymes needed for the metabolism, enzyme needed for the replication of the viral genome, as well as the transcription and translation of the viral genome, which helps in the final production of the capsid proteins. So, in biosynthesis stage, the viral nucleic acid is replicated. It also starts producing the hot proteins and the enzymes needed for the replication of the genetic material, as well as the synthesis of the proteins. In the next stage, it starts assembly or maturation. In this stage, what happens? Assembling of the viral components to form Dr. virions takes place. That means the genetic material that is replicated is getting packed up into the protein covering to form a complete mature virion. And in the last stage, that is the release stage, it liberates the progeny virions by destructing or killing the host cell. So these are the 5 major sequential steps in viral multiplication. So just to recap, what happens? First, the virus comes adsorbed or attached onto the surface of the host cell. In the second, penetration, either the virus as a whole enters into the host cell or only the genetic material is injected into the host cell. Then biosynthesis takes place. What happens during biosynthesis? The genome replicates, the protein coat is being synthesized. Then comes the assembly stage where the genetic material is packed within the protein coat, and finally the release by bursting open the host cell. So this is how virus multiplies. So a recap of the key points we were discussing today. Virus, what is virus? The term means toxin. It is, you can define a virus as an obligatory intracellular parasite with a genetic material, which can either be a DNA or RNA, and a protein coat called the capsid, which can grow and reproduce only within a living host. Then, coming on to the classification of viruses, 2 major classification systems, International Committee on Taxonomy of Viruses, ICTV, and Law of Horn and Tonia classification or the LHT system. ICTV classifies viruses into 8 clusters based on 3 major characteristics. One is the kind of nucleic acid present, whether it is DNA or RNA. Second is strandedness of the nucleic acid, whether it is single-stranded or double-stranded. And the final one, presence or absence of an additional envelope. LHT system uses 2 more characteristics, which includes the capsid morphology, like size, symmetry, and the number of capsomeres, and the range of host, along with the kind of nucleic acid, the strandedness of the nucleic acid, and presence or absence of the envelope. Then in this class, we discussed about the steps in virus multiplication. It includes adsorption, that is attachment, penetration, whereby the genetic material is injected into the host cell, uncoating, biosynthesis, where the genome of the virus is replicated as well as the capsid proteins are made, maturation or assembly, where the genetic material is packed into the protein coat, and finally release. So we stop for the day and we will discuss more on the structure of bacteriophages and the other types of viruses in the next class. Thank you.